All right, thank you for the introduction and for giving me the opportunity to talk about target enrichment for next generation sequencing. I'm going to be talking about some methods, performance metrics, and application data, but I'm going to start with why do target enrichment for next generation sequencing? Here I'm showing you a representation of the human reference genome. This is about 3.2 billion bases. The human reference exome is about 30 million bases, or about 1% of the genome. So these are the coding regions of the human genome. And then an example of a small human gene panel of around 300,000 bases, or around 0.01% of the entire genome. So if researchers are only interested in, let's say, this 300,000 bases, why would they sequence the entire human genome? So ultimately, it's to enable labs to focus on the target content of interest. It also lowers overall sequencing cost per sample. So, if you're going to sequence the entire human genome, it's going to cost a lot more in sequencing than if you're going to sequence, say, the exome instead. Similarly, it enables labs to use more or run more samples per individual sequencing run, which means you can get more done with a single sequencing instrument. And it enables deeper sequencing when detecting low allele fractions of, let's say, 5% or even lower. And in some cases, you want to go below 1%. And anytime you're going to these minor allele fractions that are very low, you have to sequence more deeply to be able to detect them reliably. Ultimately, it's all about efficiency and lowering cost per sample. So I'm going to talk about a few target enrichment technologies, hybridization capture being the first, then amplicon technologies, and then primer extension target enrichment. And this is called PEAT for short. Starting with hybridization capture technologies, we start with a DNA molecule, a double-stranded DNA molecule. These need to get fragmented, and after fragmentation, we attach universal adapters onto the ends of those fragments. This is denoted in gray, and this is so we can actually use the sequencer. So these are sequencer-specific adapters. After these libraries are made, we hybridize a probe that has a biotin on its 5' end, and these biotins will bind to streptavidin paramagnetic beads. So we hybridize these specific probes to target regions of interest. After this, they get bound to streptavidin beads, and we wash away off-target fragments. After these off-target fragments are washed away, we amplify all of these libraries. So these enriched libraries are amplified with PCR and they're ready for sequencing. So the entire workflow for hybridization capture takes about 1.5 days. So this is a little bit of a longer workflow. It's still not terrible, but it's not a one-day workflow. It offers broad panel sizes. This ranges in tens of KB to tens of megabases. So I view this as small targets to exome and larger than exome targets. And it offers best-in-class enrichment performance. So when people use hybridization-based capture, they expect to have really good performance because typically it gives very good performance. Next is amplicon technologies. So again, we start with double-stranded DNA and we use target-specific primers. This is denoted in brown. These go through PCR, so target-specific primer amplification, and usually this is limited amount of amplification, so a limited number of cycles. Following this, these universal adapters, again, these sequencer-specific adapters are ligated onto the ends. And then these libraries are amplified through PCR. So in general, amplicon technologies offer the shortest and easiest workflows, and these tend to require less than a full workday. In general, they're targeting small panel sizes, so this could be a kilobase to hundreds of kilobases. So not quite as broad as, let's say, hybridization-based capture. And then the enrichment performance is good, but not on par with hybridization capture technologies. One last thing to note, so in some cases, researchers are interested in detecting novel fusion partners. In these cases, it's like, if um, you're looking for fusion genes, and if you don't know one side of the fusion gene, you can't design PCR primers to target that. So because you don't know both sides of what you're looking for, you can't use amplicon technologies to detect novel fusion partners. Lastly is primer extension target enrichment. This is PEAT for short. So similar to hybridization capture, we start with double-stranded DNA. This goes through library preparation where we attach these universal adapters. We go through a single round of primer extension. So this is a target-specific primer. It has a biotin on it. This biotin is used to capture onto streptavidin paramagnetic beads. Gets pulled down, we wash away off-target fragments, and then we apply a second primer. We call this our release primer. It's 5' to the capture primer. When this gets extended, it pushes in a target-specific manner, so molecules that are on target, off of the bead where it can be collected, and then put through library amplification. So again, this is our enriched library amplification step. This is about a 1-day workflow. So it's a short workflow. It's faster than hybridization capture, not quite as fast as amplicon technologies. It also supports small panel sizes similar to amplicon, so kilobases to hundreds of kilobases. It offers best-in-class enrichment performance. So very similar to hybridization capture, the performances are very similar. So this is better than amplicon technologies and on par with hybridization-based technologies. And something I don't have here is that we can actually detect novel fusion partners with this technology similar to hybridization-based capture. So now I'll jump into different types of metrics. So I'll talk about on-target rate. On-target rate is a measure of the percent of reads that overlap the target regions of interest. This is what I'm talking about. So the target region of interest is in blue. This is surrounded by the green box. All of the blue lines are on target reads, even if they're not completely they're not completely inside the target region of interest. So you want more of those blue reads, less of the gray reads. The gray reads are off target reads. And ultimately, the higher the on target rate, the less sequencing that is needed. The next one is coverage uniformity. So coverage uniformity is a measurement of evenness of coverage across regions of interest and can impact the amount of sequencing needed. Better uniformity is illustrated where highest performing and lowest performing regions are closer to the mean depth. So here I'm showing an example of a better uniformity plot. The highest performing region is region A at 700 depth of coverage. Region J is at about 300x depth of coverage. And the green line there is to denote where you want to be. This is sort of your minimal depth of coverage requirements. You could see the lowest performer is at that 300 depth level. Poorer uniformity is illustrated where highest performing and lowest performing regions are farther from the mean depth. So here in the poorer uniformity, you can see region A is at about 900, region J is at about 100, and ultimately you can see it's uneven coverage. Why is this important? Well, when uniformity performance is better, less sequencing is needed to make up for the lowest performing regions. So again, let's say we want to get to 300x. In the better uniformity, no additional sequencing is needed. In the poorer uniformity, you need 3 times more sequencing to bring the low performing region to 300x. That's in J. So here's example application data from some PEAT samples. We started with 50 nanograms of low, mid, and high quality FFPET DNA. And we put this through library prep and target enrichment. On target rate is over 70% across all sample qualities. And coverage uniformity or percent basis greater than 300x is at around 99% across all of the different samples and qualities. There is one outlier in the low quality where it was at about 92%. So in summary, target enrichment is used to lower sequencing cost per sample. It allows you to process more samples per sequencing run, and you can achieve deeper sequencing when detecting low and ultra-low allele fractions. Multiple technologies are available for use. There's hybridization capture. This offers broadest panel sizes, highest performance, but it also has the longest workflow time. There are amplicon methods, which are the fastest and easiest workflows. They cover small panel sizes, but it has lower enrichment performance compared to hybridization capture. And lastly, there's primer extension target enrichment, or PEAT. This is fast and easy, not quite as fast as amplicon methods, but still can be done in about a day. Small panel sizes are supported, but the enrichment performance is on par with hybridization capture, kind of best in class. With that, I'd like to thank you for your time.